as our today's seminar speaker. Dr. Lee received a Bachelor of Science in Engineering and a Master of Engineering degrees with a nuclear engineering emphasis from the University of Florida. He then received his Doctor of Philosophy degree, also with a nuclear engineering emphasis from the MIT. Following postdoctoral appointments at Los Alamos National Lab and the University of Maryland, he started his career in therapeutic medical physics. He's an expert and former trainer for the CyberKnife that I'm sure you all know about, the robotic radiosurgery and radiotherapy system, having supported institutions including Georgetown University, Erasmus Medical Center, the University of California, San Francisco, and Fox Chase Cancer Center. His roles in medical physics have included staff and lead hospital physicist, owner of a medical physics consulting company, senior director of global medical physics for Accuray Inc., and director of clinical development for TAE Life Science. His research interests include accelerator-based boron neutron capture therapy, bnct, stereotactic radiotherapy, and development of quality assurance tests for bnct. So I'm personally a fan of bnct because it was my thesis topic. So I'm looking forward to listening to this talk, and whenever you are ready, feel free to go ahead, Chad.
Okay, thank you so much, Angela. My PhD thesis topic was also accelerator bnct. I thought 20 years ago that I was done with bnct, and life has a funny way of taking you places you didn't expect. So it's pretty exciting that we're now in a position where bnct is being talked about, and not just talked about, but we are now installing accelerator-based bnct systems around the world. And it is, I think, going to have a really big impact on cancer therapy, fundamental approaches. And let me dive in and share my screen. I'll start my PowerPoint.
Yeah, I can see you well.
Wonderful, thank you. So given the fact that we have a lot of nuclear engineers attending this talk, I decided I wanted to give you a taste of a lot of the complex things about bnct, boron neutron capture therapy, that are distinctive from other types of radiation therapy, and why we have myself as well as a full-time nuclear engineer on our staff, and are hopefully looking to expand that to hire another nuclear engineer. It is one of the only therapy areas where you have this interesting mix of radiation transport, shielding, unique shielding issues with neutrons and photons, that you have radioactivity, you have activation and storage of radioactivity. It combines things into a real interesting mix, and even a little radiobiology gets mixed in there. So my apologies, I don't know the background of everybody, but I wanted to give a little context on traditional radiotherapy. High energy X-rays are the standard way to treat cancer. You identify a tumor using a CT and or MRI or PET scan with pretty good resolution, and then you assume there's no physical changes from the time you take the CT until you deliver the treatment, and your accuracy is, this is all geometric. It's based on the resolution of the image, the assumption of no changes, and then you aim beams of high energy X-rays at the location that you've identified on your CT. So precision in your equipment becomes very, very important for traditional radiotherapy. You also create high levels of radiation damage at the tumor location by overlapping a large number of beams. So the idea is that a single X-ray beam is not going to create enough damage to the tumor to be able to kill it, because, as you can see, other parts of the body are going to experience dose behind the tumor, or even ahead of it, in front of it, depending on the direction that the beam is coming. And so to get high levels of dose, you actually use a geometric approach. You overlap And your goal really with radiotherapy is to deliver enough dose so that you have double DNA strand breaks. You want the DNA strands to not be able to replicate anymore. You're not melting the tumor, you're not blasting the tumor, you're stopping the tumor from growing. And so it all comes down to how efficient is the radiation at doing this damage to the DNA. And for X-rays, it's pretty inefficient. The process is that the X-rays undergo typically Compton scattering off of the electrons in the body, and those electrons, as they then take this kinetic energy and are traveling through, produce reactive chemical species, free radicals, etc. And these free radicals are the things that typically do the damage to the DNA. Very rarely is DNA damage directly from the X-ray interaction. It is from this chemical process. So the effectiveness of the radiation in damaging tumors is dependent very much on the density of these reactive chemical species that are produced. And heavy charged particles like alpha particles create more damage from the same dose because they create higher densities of these free radicals. So what is boron neutron capture therapy? How is it different? So the first step, it's a combined therapy. There is a pharmaceutical aspect and a radiation aspect. And unless you have both together, you don't have the bnct effect. So the first step is that the patient is given a boron pharmaceutical. It is non-radioactive. So if the patient, it has this infused into their body and then no radiation is delivered, they'll just pass the pharmaceutical out of their body and there won't be any problem. It's non-toxic. The reason it's labeled with boron is, as I'm sure all of you know, boron 10 loves to soak up thermal neutrons. So, and when it does, it splits into an alpha particle, and the residual lithium 7 nucleus is sufficiently light that it actually acquires an appreciable fraction of the Q value of the reaction and has a decent amount of kinetic energy. So both the alpha particle and the lithium nucleus go flying away from the location of the reaction, but they only travel, because they're heavy, massive charged particles, they only travel about 5 to 10 microns, which is roughly the dimensions of a human cell. So what this means is that if you've designed a boron pharmaceutical that goes only into the tumor and not into the surrounding healthy tissues, then when you aim a beam of neutrons at it that thermalize at the location of the tumor, you're going to get a very high amount of energy deposited, and it's only deposited in the cells that had high concentrations of boron to begin with. So the pharmaceutical is critical to having a very effective bnct process. The other key thing is not only is that high amount of reaction energy deposited only in the cell, because it's an alpha particle and a lithium 7 nucleus, these are heavy charged particles, and therefore they are creating this high density of the reactive chemical species. The key thing here is that the precision is all the way down to the cellular level, and it's driven by the biology, not the geometry. So here's another slide showing what I was describing. The key is that, remember that the boron 10 is splitting into 2 heavy charged particles that are therefore high LET. I'll remind everybody what LET stands for, but that's pretty critical to why it's more biologically effective than traditional high energy X-rays. And the other key point being the pharmaceutical drives the precision of how the treatment works and how accurately you can differentiate between tumor and healthy cells. One thing I will mention on this slide is you see the patient line here on the left, and they have what's called a neutron generator, but you have a proton beam coming through. So the way traditionally bnct was produced was to use a nuclear reactor to produce an intense neutron field with the correct energy spectrum And that limited where you could do this, right? It was primarily at universities that had research reactors, as well as hospitals that wanted to get involved in this. What we're doing now is what's called accelerator-based bnct, where you direct usually a proton beam. It could also be a deuteron, but primarily proton beams onto a target that undergoes PN reactions that converts the protons into neutrons. So bnct, there's three key aspects that you need to make it work. You need to have good pharmaceuticals. You need to have an efficient proton delivery system and target system to convert those protons into neutrons. And then you need to have all the other components that are always necessary for treatment planning. You need to have a dose engine to be able to calculate how these different components of radiation travel through the body. You need to have a robotic couch to set the patient up effectively. We are the only, just a quick plug for my company, TAE Life Sciences, we're the only company that's doing all three of these. Half of our expenditures each year go towards developing next generation drugs, because the higher the uptake in tumors, the shorter the treatment time. The higher the differential between uptake in tumors versus surrounding healthy tissue, the more effective you're going to be at protecting healthy tissues. So we do a lot of work on all three aspects of the delivery system. And this last portion, the components that allow you to fit into a hospital radiation oncology environment, is really what I do as Director of Clinical Development. So, as I mentioned, BNCT is really concerned about, well, not just BNCT, but all radiotherapy is really concerned with trying to get as many double-strand DNA breaks as possible. Here we see a normal DNA strand, and you can have single-strand breaks, but enzymatic repair is actually very effective at repairing single-strand breaks. Even if you have two single-strand breaks on either side of the chain, as long as they're far enough apart, they're really treated as two independent breaks, and it's not a problem. A double-strand break is actually very difficult to repair, and it must be within about 30 base pairs to be considered a double-strand break. So what can lead to a high percentage of double-strand breaks? The key is in the linear energy transfer of the particles that are, or the radiation that's being delivered. It's a measure of the ionization density along the radiation path. Hopefully everybody's heard of LET in this audience, so I don't have to go into too much detail. But what I show here in the figures is a low LET photon cascade on the left, and then on the right you see an alpha particle. So just to illustrate the increased density of interactions and ionizations, and therefore higher density of these reactive chemical species, the free radicals that are going to attack the DNA. And what creates these high densities? Heavy particles, typically charged particles, alpha particles, protons. We list neutrons in there. The reason we do it is that neutron interactions, and we'll see this later, are going to typically produce heavy charged particles. So the neutron might scatter off a hydrogen and produce an energetic proton, which is high LET, or it might undergo an in-alpha reaction, like with boron 10. Now you've got something that's high LET. So we classify neutrons as high LET, even though they are not a charged particle. Another thing that hopefully you've seen before that we talk about a lot in BNCT is RBE, the relative biological effectiveness. And we want to get, RBE is necessary when you want to compare different types of radiation at the biological level. What is the impact biologically that's happening? It's real straightforward to measure what the absolute dose is, what the physical dose is that is deposited by radiation in tissue. That's fairly straightforward, regardless of the type of radiation. The question is, if I deliver the same number of grays from X-rays as I do from neutrons of a certain energy, am I going to get the same biological effect? And the answer is no. So RBE is a way to try to get at an understanding of what, how to quantify that in some way. So for a cell survival curve, which hopefully you've seen, RBE is defined by taking the curve for a default reference low LET source of radiation, typically 250 kVp X-rays, and you get a cell survival curve like this. Notice that what we're seeing here is a linear axis for dose and a logarithmic axis for the surviving fraction. Now you take the dose, the radiation source that you're interested in, which I'm showing in red, and you repeat the experiment. And what you then do to determine an RBE is to choose a biological endpoint. In this case, let's call this 1% surviving fraction. And then what you do is you find the doses that correspond to that biological effect, and the RBE is taken as the ratio of those doses. So it is a way of getting at what biologically is happening. The reason this is important is that because we have a mixture of radiation sources in bnct. We have the neutrons that are going into the patient. We have the alpha particle and lithium being produced in the reaction. We also have the neutrons undergoing all kinds of reactions, including elastic scatter with hydrogen. You've got capture reactions in nitrogen. You've got capture reactions by the hydrogen when they thermalize to produce gamma rays. So you also have gamma radiation. So when you've got all these different components, and given the fact that they all have different LETs, they're going to have different RBEs as well. So we want to actually use RBE as a way to try to create a level playing field to do our dosimetry. So what I'm showing here is that the reason I mentioned LET and the reason I mentioned RBE is that they're related. So on the x-axis here, you have LET, which is, if you notice this time, this is the logarithmic scale. So we're going over a very wide range of LET values, and this is a linear scale for the RBE. But what's interesting is that there's a peak. And regardless of the biological endpoint that you define, the peak happens right around 100 keV per micron LET. And on the next slide, I'll explain why. The key here is that LET is not the same as kinetic energy. As your kinetic energy decreases, as heavy charged particles are slowing down, the LET actually goes up, and then it drops back down again. Luckily, part of the reason that bnct works is that for the boron-tin reaction, the two products, the alpha particle and the lithium-7 nucleus, are born with kinetic energies that correspond to LETs around 150 to 200 keV per micron near the peak. So it's not just that these are high LET particles that are produced, but they're the most biologically efficient that you can hope for. Compare that to the RBEs down here for photon sources, cobalt-60, gamma rays, or X-rays down here. And you can see this means that I can give, if we're looking at this biological endpoint, this means that I could give 7 times less physical dose to have the same biological endpoint. Or conversely, if I give the same physical dose, I should be 7 times more impactful biologically. This is a real key to bnct. So the question though is, why is there a peak? Why is it that for low LETs it's not very effective, but then high LETs aren't effective either? And this is trying to explain this a little bit. For low LET, what happens is primarily single strand breaks. You don't typically get double strand breaks because of the low ionization density. And so in order to have cell killing, you typically have to have interaction. You have to have a higher dose in order to be able to get to those double strand breaks. And so it's just not very effective. And that's why we say that X-rays are biologically inefficient. As the LET increases, the ionization density increases, and you can get a very efficient series of ionizations all the way along radiation track, which produces a very efficient density of free radicals that can then cause damage to DNA. And so it's very biologically efficient. But if you go to even higher LETs, what happens is that it's still very efficient, but the problem is it's so efficient, it's overkill. You're producing more, you can only kill a strand of DNA once. Once it has a single double strand break, it's unlikely to repair. So if it undergoes 5 double strand breaks, it's overkill. You've done more than you need to. So you end up not being as efficient because you're kind of wasting your effort with the higher LET radiation. So that's why you get this peak that we're seeing here. So how do we actually produce the neutrons? We have an accelerator called a Tandem accelerator, which you see here. And then we have a high voltage supply and a source of negative hydrogen ions from this platform. This platform accelerates the hydrogen negative ions to about 150 keV before injecting them into the Tandem accelerator. And on the next slide, I'll show you how it's designed. They are converted to protons in the Tandem and also accelerated to 2.5 MeV energy. It's a very high current, 10 milliamps, and those protons are then transported along beam lines directed with magnets, and we can treat up to 3 different rooms from a single accelerator. And these proton beams are then directed to a lithium target. Lithium 7 in particular has a very efficient PN cross-section at producing neutrons from protons. You can see inside here, we've got a series of shells, conducting shells, that produces a voltage gradient across the Tandem. And in the center of the Tandem, we have a charge exchange target. So we have negative hydrogen ions, a hydrogen with 2 electrons entering from one side. Because of the negative charge, it gets accelerated to be about half its desired energy when it gets to the center. But now when it passes through this argon gas chamber, it strips the electrons off the protons, leaving now a positive proton. And so now because you've had a charge flip from a negative to a positive, then the very voltage gradient that was drawing you in to the center is now pushing you out on the other end, and also doubling the energy gain in that process. So it's a very compact, very efficient way to get us to this 2.5 MeV beam. Also, it doesn't have, there's no gears and pumps and what have you. We have to maintain vacuum, but all of this process is done electrostatically. So it's also, that makes it tunable, it makes it robust, and makes it fairly inexpensive. So we're pretty proud of this design as a way to produce our protons. Then we aim these at a lithium target. The lithium does not need to be more than 100 microns thick. Once you go past 100 microns at 2.5 MeV, you've slowed down past the threshold energy for the PN reaction in lithium 7. So you don't gain anything by having anything thicker. And lithium is very hard to cool. It has a melting temperature of only 180 Celsius. And we're dumping about 3 megawatts per square meter onto this target. And so keeping that from melting is a real challenge. So we actually have a copper backing with this thin 100 micron layer of lithium that you see here. And then the proton beam is rastered over the surface to get a uniform broad beam of neutrons that are produced. So we have to figure out from the protons coming in what the energy spectrum and the angular distribution of the neutrons coming out will be. And this is our thick target yield equation. It shows the yield as a function of neutron energy in the lab and the scattering angle in the lab. And these are constants. This is the differential cross section for the PN reaction in the center of mass frame. And it is a function of the proton energy coming in. So you have your differential cross section here. This is the stopping power for the protons in lithium. And this is a Jacobian. This is actually converting you from one lab space to another. This is actually a thick target yield. This is an integral equation. Why is there no integral in here? And it's because there is a one-to-one relationship between proton energy and proton and the scattering angle in the center of mass, and neutron energy and the production into the lab. And so you can go between these two. There's a delta function essentially that occurs because of this one-to-one relationship, and it collapses an integral down into just a point-wise calculation, which makes it very efficient. To get some idea of what this Jacobian looks like, this isn't the value of the Jacobian. The colors are simply trying to show how the neutron and lab space gets twisted using the Jacobian and the kinematics of the reaction to get you into what proton energy and center of mass angle you want to look up from your cross-section data. So what we are going to find is that for a 2.5 MeV proton, that's going to produce neutrons up to about 780 keV. We don't want neutrons higher energy than that because it produces too much skin dose. So we want to limit that amount. But if we then look at this range of mu being the cosine of the angle from 180 to 0 degrees, and over this energy range, if I'm curious about the yield in, let's say, this square here, that would correspond to, this square would correspond to, I believe, this square here. It's not a square anymore, but this region. And so it's a fairly complex, it's not an integral, but it's a fairly complex transformation that has to be performed to calculate the yield. This hopefully gives some idea of the lab angle from 0 to 180 degrees. This is the angle of emission from the target and the neutron energy that corresponds to it. And this shows, for example, 2.5 MeV is the top curve. When I have a proton initially striking the lithium with that energy, if it undergoes a reaction, I can use this to figure out the one-to-one relationship between the angle of emission and what the neutron energy would have to be. And then I can use the cross-section to tell me the yield at that point. As the proton beam passes through the lithium, it will slow down through all of these energies until it passes through the threshold energy, which is about 1.88 MeV, and cease to produce neutrons. So we have a very complex yield calculation that we've now standardized and use that in our Monte Carlo calculations of dose. I think I have about 10 minutes, so I'll try to get through this next stuff fairly quickly. As I alluded before, we have a lot of different sources of radiation and processes. Just the neutrons alone are undergoing scattering reactions, radiative capture, and different nuclear reactions. And they're all pretty important to what we're looking at. Elastic scattering with hydrogen is the key dose component for faster neutrons at the higher end of the range. And of course, those protons that are produced are high LET. But we also have hydrogen undergoing thermal neutron capture, and that produces a 2.2 MeV gamma ray. That's pretty intense. And so the whole head is bathed with gamma rays produced by the neutrons thermalizing and interacting with hydrogen. You also are producing, the body is not water. The body has a lot of trace levels of sodium and chlorine and calcium and phosphorus and other elements. And sodium, for example, can produce a 7 MeV prompt gamma. So you have a very complex spatial and energy distribution of gamma sources that are produced by the neutron beam. Also, there's a fairly small percentage of nitrogen in the body. It's on the order of, I think, 1 to 3% at most. But for thermal neutrons, it dominates the dosimetry because of the NP reaction that produces carbon-14. It produces a 600 keV proton. Now we're back to high LET land again. And so it's going to be a very key component when you calculate the dose of thermal neutrons, what dose components the thermal neutrons are producing. So, and then of course there's the boron tin reaction, which is the whole basis for the treatment. So it's a very complex mixture of radiation sources. I don't have time to go into many details of our calculations, except to say that we currently are using MCNP as our baseline for neutron and gamma transport. As we move into a commercial product, we can't be using MCNP anymore, but we will still be using Monte Carlo calculations. And this is an example of a facility layout for a customer coming up, where this is the accelerator room. Above this square is where the tandem accelerator would be located, sending the proton beam down here. And we have 2 treatment rooms. We have one over here, sorry about that. So you can divert the beam to be aimed at this room, or it can go down here and be diverted and be aimed at this treatment room. Each room has a short maze, as well as double doors, shielding doors. And then this is actually an alcove with an elevator, because the PN reaction on lithium leaves behind a radioactive beryllium-7 nucleus. So this elevator takes you to a storage room for use targets. So we also in this room have a patient phantom sitting on a robotic couch right up against the beam port for our room. So this is what was used to perform our model. We used the calculations I showed before to get our neutron source, and then calculate through all of this equipment. We have something called the Beamshaping apparatus that takes the raw neutrons and converts them into a spatial distribution and energy spectrum that we want for a treatment beam, and then model the radiation throughout the facility. So this is an example where we sent a proton beam down into to treat this patient in treatment room 1. And you can see there, we have here dose rate from neutrons only, and here we have a combined dose from all the different radiation sources, including all of the prompt photons that are produced. And you can see, for example, that our shielding is doing a pretty darn good job of containing the neutrons, the neutron dose, but the prompt gammas that are produced from the neutrons end up producing some sprays of radiation that go into the other treatment room. So based on this calculation, we have to add shielding here and here so that we can protect this back spray effect between rooms. So this is bread and butter nuclear engineering calculations. Very cool stuff. I think I'm on my last slide or next to last slide, so I think I'm on time. I already mentioned that the target itself produces beryllium-7 as a radioactive byproduct. But as we all know in nuclear engineering, neutrons will activate. It's just best to assume that everything's going to get activated, and then it's just a question of how much, how quickly, how long is the half-life, and what are the characteristics of the decay radiation, and how do I then protect against it? So something in addition to all of the design work on the radiation source itself, we are involved in every single element of design for every component in the facility, because all it takes is a very small amount of something with a fairly small neutron flux to produce something that will become radioactive and just build up over time if we're not careful. So I show here aluminum. If you have neutron capture of aluminum, it produces something that's pretty intense in terms of the decay radiation, but its half-life is only 2.2 minutes. So we have a lot of aluminum on our machine. We use aluminum as much as possible because even though it gets hot, once we turn off the beam, it's going to essentially be gone in about 15 minutes. So aluminum is our friend. The copper backing that we need for our target is very important for thermal management of the lithium to make sure it doesn't melt. But the problem is the copper will capture neutrons. Copper 63 in particular has a pretty small cross-section for absorption. And it's interesting because it has a combination of internal conversion and positron emission. So its decay profile is pretty interesting. But it's not that bad to shield against, quite frankly. The key, however, is that it's got almost a 13-hour half-life. So it will build up over the course of a treatment day. So that's a challenge. That's a challenge. And then I included manganese here simply because manganese is a trace component in stainless steel, most stainless steel types. So there's a very small percentage of manganese in these components. It's not a huge cross section, but the half-life is about 2.5 hours, long enough that it will build up over the course of a treatment day. But it is incredibly penetrating. It produces so much decay radiation that small amount dominates. Once that aluminum decays away, it's manganese dominating, even though you have a small trace amount of it. So we have regions of the facility where we don't allow stainless steel. Even for bolts and nuts that we're using, we are now having to switch to things like titanium because of these properties. So it's pretty cool that nuclear engineering is able to affect really all the way down to the nuts and bolts, literally, on the machine and what we're using where. So that was very, very rushed. Went through a lot of information in 40 minutes. I hope that it was somewhat comprehensible, and I am open to any questions you have.
Yes, thank you very much, Dr. Lee, for the presentation. I think it was great, and it covered all the different aspects and really conveyed how bnct is a multidisciplinary type of treatment. So now I invite anyone who has any questions to unmute yourself and feel free to ask the question.
Can I have a question?
Sure, go ahead, Xiaochun.
Thank you for your very informative presentation about this frontier technologies. So I have two questions. The first is that when you are doing the multicolor simulation using MCMP, how are you going to deal with the boron concentration changes as you irradiate the neutrons? And the second question is that, is there any way for now could be for you to online monitoring the boron concentration in tumors and then in the normal tissue? Thank you.
Wow, that second one's a very tricky question. So let me start with the first one. So the boron pharmaceutical, when the patient is infused, has a fairly rapid change with the BPA is the most common pharmaceutical that's used. It's been in use for several decades. And unfortunately, after infusion for a couple hours, it has a pretty rapid decay until it goes to a very gradual decay about 30 minutes after infusion stops. So there's, as you mentioned, that's just in the blood, but there are concentration changes in all of the organs over time. So when we're doing modeling, what we do is we assume a fixed concentration of boron. So that we essentially what we do is we divide up our dosimetry into different components. So we have the dosimetry, we have the dose distribution from the boron components only, and we do it as a function of concentration. So it might be gray equivalent per ppm of boron 10. Then we can take that from a basic neutron level and we can scale it according to the concentration and then integrate that over time to get what the total dose would be. One key point, and you've kind of hit on this, when you do your treatment plan initially, you assume a concentration and you come up with how long the beam should be left on. On the day of, we will actually take measurements of the boron in the patient's blood and use that to correct the treatment plan. And we correct that with both the concentration change and also the time dependency that is assumed. But that's a key word. There are some assumptions with that approach. We are assuming that there is a constant relationship between the concentration of boron in the blood and the concentration in tumor, and between the blood and other tissues. The reason we're doing that is because we do not currently have a good way to do in vivo dosimetry of boron, which is your second question. So the approach that most people assume will be most effective is prompt gamma imaging. So with the boron 10 capture reaction of thermal neutrons, about 94% of the reactions produce a 478 keV gamma. It's a 478 or 431. It's one of those. I get them mixed up. My apologies. But it's a distinctive gamma ray energy. And so what people are looking at is there a way to actually do spectroscopy on that signal, and from that quantify the amount of boron in the patient? There's been lots of papers written over the last few decades on prompt gamma imaging for bnct. Nobody has it as a product yet. It is very challenging because of the discrimination that's needed. You're talking about only a few parts per million of boron, even with a tumor that's very efficient. And you've got all these other signals coming from everything else going on in the room and in the body. So that is actually something we're working on right now. We have a project with a physics professor at MD Anderson that's working on designing detectors for that very use. And I'm involved with a few other groups that are also looking at other approaches. So that's going to be pretty crucial for us to be able to better estimate the dose that's delivered from bnct. Good questions.
Thank you.
Great. Do we have any other questions? Perhaps we have time for one or two more questions. Well, if not, I will ask one. Since we focus in this talk on the technological aspects, I wanted to ask a question more on the clinical side. The question is, the energy of the beam is also strictly related to the type of treatment that can be performed. So which treatment are you currently targeting the brain tumors, the melanoma has had a good outcome for bnct. What is the plan for that?
Yes, so we are expecting that our first 3 targeting areas will be head and neck tumors, glioblastomas in the brain tumors, and melanomas, as you mentioned. And what's really driving that more than anything is the pharmaceutical. BPA is designed specifically, it uses what's called a LAT1 transport mechanism, and all 3 of those cancers use LAT1 to a high degree, so you would bring in a lot of BPA, and therefore your concentrations would be higher. With our pharmaceutical development side of things, we are looking at other transport mechanisms that would work with other types of tumors. So for example, for a lung adenocarcinoma, we would look at the transport mechanisms for that and design our pharmaceuticals to be taken up with that. And so, and then once we have some idea of how much boron is taken up, then we can design our neutron beam energy spectrum to, the key is that you want it to be high enough energy so that it travels deep before it thermalizes, but you don't want it such a high energy that the skin dose is too high to offset what you're doing. So that effect is dependent on the location of the tumor, the depth that you want, as well as how much boron you can get in that location. So it's actually a synergistic development with our pharmaceutical team. So they are looking at, I want to be very careful what I'm saying here. I think it's safe to say that things in the thorax and also like sarcomas, I think would really benefit. Part of the reason is because sarcomas are traditionally considered radioresistant. And the reason for that is they don't respond well to low LET radiation. So high LET is perfect for them. Same thing with melanomas. So and the nice thing is a lot of sarcomas are fairly superficial. So you don't have to be able to penetrate as much to treat those. We are making sure that our treatment robot has both a couch so the patient can lie supine or prone, but also has a chair so that the patient could be sitting up and we could have access to, say, their back or their chest. So we're working on that. I think that is a bit down the line because when we get our first sites installed, which will be within the next few years, we'll start clinical trials. And we have to do clinical trials for our initial drug, which is likely to be BPA, because there's previous studies and we don't have to go through all the toxicity phase 1 studies. Before we can do it, we need to do all of that before we can get FDA approval to do this as a combined therapy, which then allows us to actually have our customers get paid for it. So there's a lot of work we have to do just for those initial 3. So we're definitely doing the work, but it's years down the line.
Right. Yes, and another advantage of bnct is that it works well for all those tumors that are mixed with healthy tissue. So those cases certainly apply. So I'm glad to hear that you're getting close to the clinical phase. And we have one last question that is from Philip Tanton, who's asking, what are the advantages of bnct over proton beam therapy, as it seems that they both require similar accelerator infrastructure?
Good question. So yes, they do require similar infrastructure. Proton therapy, all radiation therapy except bnct uses the radiation source as the cell damaging component. So, sorry, okay. So if you need to treat a deep-seated tumor with proton therapy, you need a high energy beam because protons lose energy very rapidly in tissue. So proton energies are on the order of 150 to 300 MeV. So what you're talking about is the same, it may look like similar infrastructure, but we are much more compact. And so you have much a lot of space reduction and cost reductions relative to what a proton can do. However, there's BNCT still, after decades of being around, long enough for Dr. DeFulia and I to actually get our degrees some amount of time ago. There's not a huge amount of clinical data on it, but proton therapy has a lot. So it's actually something that's very predictable. Keep in mind that with the proton beam, all the exciting stuff is happening at the Bragg peak. So you're sending in your protons, and then you've got your Bragg peak with high dose deposition. And so in order to deliver appreciable dose to a tumor and not to the things around it, you raster this beam. There are other approaches, but I'm just going to talk about rastering for now. You raster the beam across a plane, and then you adjust the energy to shift the Bragg peak, and then you fill in the volume that way. You're right back to needing precision, which means you're only as accurate as your imaging and your setup immobilization. BNCT gets away from that criterion. As long as you can get a broad beam of neutrons aimed at a patient that has this pharmaceutical, the pharmaceutical does the high precision work. The last thing I'll mention is that while protons are high LET, they are not very high LET. So the average LET used for proton therapy is only 1.1, whereas the LET for the alpha particle and lithium-7 are on the order of 3. So you get much more biological oomph from the BNCT reaction than you do from protons. So I gave a few different differences there. I hope that's enough.
Yes, thank you so much, Dr. Lee, once again for speaking to us today. We really enjoyed your talk, and we thank you again for being here.